Hello, my name is Lillia Dahlstrom, and I am a freshman honor student participating in the First Year Honors Mentor Program, and I will be sharing my research with you today. My research project was involved in the Department of Natural Resources and Ecology Management. My project is called Investigating the Biomethane Potential of Various Prairie Plant Species. Using samples of previously collected prairie plant species, I am testing the relative biodigestibility of different prairie species under different cultivation methods. My goal is to determine which prairie plant species has the highest biomethane potential so we can maximize the amount of biogas we can generate by planting and processing more of that specific species. This project was under my mentor, Dr. Lisa Schulte-Moore, who is the principal investigator for this project. I also worked in the lab with Almin Rahik, who is a PhD student. Before I begin describing the process of this project, I will give you some background on some elements in the terminology I will be using. Prairie strips are strips of land where native prairie plants have been planted on agricultural land. These prairie strips are tools for conservation that have multiple uses. They are an environmental tool that can be utilized in different ways. They can be used to improve the health of the environment by helping to prevent runoff from agricultural land while providing habitats to native wildlife. These strips are being utilized in several agricultural landscapes, and the prairie material can be harvested to create biogas from anaerobic digestion. The process that I utilized in my project that transformed the prairie grass into biogas is called anaerobic digestion. Anaerobic digestion is a process in which bacteria break down organic matter, such as animal manure and wastewater biosolids, in the absence of oxygen. This process of creating biogas starts by putting biomass into a reactor. These reactors contain microbial communities that break down and digest the waste and produce biogas in digestate, which is discharged from the digester. Biogas is composed of methane, carbon dioxide, hydrogen sulfide, water vapor, and trace amounts of other gases. Biogas is purified by removing the low-value parts, like CO2, to generate renewable natural gas. The energy in this biogas can be used like natural gas to provide heat, general electricity, and power cooling systems. This can be sold and injected into the natural gas distribution system, compressed and used as vehicle fuel, or processed further to generate other advanced biochemicals. These are the prairie plant species that I used in this project as feedstock for anaerobic digestion. So I used wild bergamot, big bluestem, switchgrass, Indian grass, round-headed bush clover, pale purple coneflower, and oxeye sunflower. All of these plant species were collected from an ISU research farm and are the most common plant species that were found in the prairie strips. For this experiment, some of these species had both a fertilized and unfertilized version, but some only had one version. The goal for this experiment is to investigate the biomethane potential of different prairie plant species in order to maximize biogas yield. My prediction for this experiment is that different prairie species will have different levels of digestibility, and prairie species from the fertilized versus non-fertilized prairies will also have different levels of biodigestibility. The data from this experiment will determine which species has the most biomethane potential. Here are the bottles of inoculum that were used in this experiment. We incubated them for several weeks to degas them, then we grinded up the plant samples and added an equal amount to each of the bottles to begin the experiment. The bottles were incubated at 37 degrees Celsius for this experiment. Each condition had 3 replicates, and I also included 3 blank bottles with no prairie grass added as a control. This process that was conducted is called a biochemical methane potential test. So this is the process that I used to collect my data. About twice a week, I regularly measured the biogas that each bottle produced using a water displacement method and recorded the amount of biogas that was produced. As we got further into the experiment, the amount of biogas each sample produced per day lowered. This is because the feedstock energy was being used up. Then, I used a gas chromatography machine to measure the concentration of methane and carbon dioxide in each of the samples. This machine works by taking a sample from the bottle and running it through a column that separates the methane and carbon dioxide particles in the sample to determine the total quantity of each. The larger quantity of methane per sample, the better for this experiment. So here is the data that came from this experiment. My prediction for this experiment is correct. Different species of prairie plants do have different levels of biodigestibility when using anaerobic digestion. This graph shows a comparison of total methane production of each of the unfertilized species. We can observe that the grasses showed a higher methane production than the forbs, which are the three on the bottom. And then here is the data from the fertilized prairie species. It looks pretty similar to the unfertilized species, although there does seem to be a smaller gap in terms of methane production between the grasses and the forbs. So these two graphs represent the raw data that have been collected from this experiment, and this graph shows the data when the methane production from the blanks that had no prairie plant mass in them have been subtracted. So this is the total amount of biomethane that each prairie plant produced. From this graph, we can see the pattern where the grasses did end up producing more methane than the forbs. According to this graph, there doesn't seem to be a correlation between the fertilized and unfertilized prairie grass in terms of the amount of methane produced. So whether or not the prairie plant was cultivated in a fertilized plot or not does not affect the amount of methane it produces. From this graph, we can see that the prairie plants that ended up creating the most methane were big bluestem and Indian grass. So for future applications for this project, the data from this experiment has determined which species has the most biomethane potential. This information will influence how seed combinations for prairie strips will be made for farmers to plant, which will allow for maximum biogas production when harvested. Utilizing the prairie grass on these strips makes this process even more efficient and profitable for the farmers and best optimize the amount of methane generated from the prairie strips. I would like to thank my mentor, Dr. Lisa Schulte-Moore again, for helping me along in this project, and Elmine Rahik for assisting me in the lab. I would also like to acknowledge that this project is part of Sea Change Grass to Gas project, which is supported in part by the Sustainable Agriculture Systems Grant from the United States Department of Agriculture National Institute of Food and Agriculture. They are the source of funding for my research materials, and this research project was made possible by them. This concludes my presentation, and I would like to thank you for watching. This research project has been very rewarding, and I hope that these conclusions will aid in any future endeavors to optimize biodigestibility of prairie plant species.